Francesca.
Thank you very much, Lorenzo, for the nice introduction and also for the kind invitation. It's a pleasure to present our activity after all these very nice talks and contributions today at the International Day of Light. Indeed, the title of my lecture would be Imaging Matter at the Electron Timescale, but I wanted to add light for imaging matter at the electron timescale, because the key ingredient here is really the light source we work on to be able to capture indeed these ultra-fast dynamics. So let me mention that we are located in Hamburg, as Lorenzo Valde was anticipating, and Hamburg has been, it's particularly famous to be hosting a very big harbor. And therefore, light sources in Hamburg have been always a big topic. And in the past, of course, this was mostly in the development of advanced lighthouses. So that was a lot of engineering for several centuries in order to aid basically the shipping to be successful in the harbor. So now there are still over 40 active lighthouses along the Elbe, which is the big river that it's bringing the big ships here to Hamburg. But the most advanced light sources are no longer lighthouses, as you may imagine. And it was already nicely covered this morning by Massimo Altarelli. Indeed, we are hosting very advanced light sources that are shining nowadays in Hamburg. So we have a synchrotron, a Petra 3, and a flash, which is BUV Softix free electron laser, hosted at DESY. But we also have the European XFEL, which is an X-ray free electron laser, a super bright X-ray laser, which is, of course, advancing the frontiers of the science we can conduct using these advanced light sources. I would not cover the science, which is investigated using the free electron lasers, as this was done already in the morning, but I would instead present tabletop light sources, which are in fact driven by conventional laser sources, and that are meant to push for extremely short pulse durations, in fact, in the attosecond time scale. And these are developed within my group at DESY. Here you can see some pictures of the laser sources and the vacuum chambers that are hosting actually our attosecond sources. So let me start by discussing about the timescales in matter, which is the key ingredients here. So as we zoom more and more into matter, as we are dealing with higher energies or higher energy scales, and this means, of course, faster and faster mechanisms. So it's well known that, for instance, structural dynamics in matter, so for instance, molecular vibrations or change in structure are happening within a time scale from the picoseconds down even to the femtosecond domain. But when we talk about electrons, then we basically move from the milli-eV energy scale up to the electron volt and even kilo electron volt scale, depending on the electronic states that are into play. And this means that we are indeed looking at faster processes from the few femtoseconds even down to the attosecond domain. If we would be able to follow even more inside matter to the nucleonic motion, then we would need even zeptosecond sources. But for today, we are limited to accessing the electronic motion, and meaning to the attosecond timescale. Just to give you an idea why it's important to follow dynamics of matter over the entire timescale, I would like to bring up a very nice example. This is actually the DNA photoreactivation mechanism. So as most of you know, when we are exposed to UV light, which is penetrating our atmosphere, we could get damage of our DNA. And in fact, this is, of course, the primary mechanism responsible for the development of skin cancer. And in fact, the damage relies in the formation of a covalent bond between two thymine nucleobases. And this is actually interrupting the nice sequence of our DNA and creating the genetic miscoding. There is, in fact, a photoreactivation mechanism which is protecting us from this damage. And again, it's activated by light, and it's operated via a photolyase enzyme, which is schematically represented here. And the photoreactivation mechanism is, in fact, based on a chain of events, starting again from the absorption of light from the HDF protein complex. And the first mechanism happening is, in fact, an ultra-fast electronic rearrangement. This intercoulombic decay is, in fact, responsible for a transfer of energy to the second part of the enzyme, which is the FADH charge donor complex. And as a result of this energy transfer, an electron is then finally transferred to the pyrimidine dimer. And in fact, this charge transfer is responsible ultimately for the breaking of this covalent bonding and therefore the restoring of the DNA sequence. This is just to transfer the information that, in fact, there is an ultra-fast chain of events, which is ultimately leading for a very complex photobiological process, the DNA reactivation. And it all starts with a purely electronic mechanism. So it's really important to follow all the time scales, so to say, and all the processes from the faster processes that deal with electrons, purely with the electrons, up to the structural changes that molecules undergo at later times. The way we can explore the ultra-fast time scale is to use, as it was already mentioned in the previous lectures, is the use of ultra-short light pulses. We need, in fact, light pulses which are as short as the physical mechanism we intend to follow. And the mainly used approach for performing a time result measurement is called pump and probe, where we use the first pump pulse to excite our target, our sample. So we bring the sample into some excited state, and then we probe the following dynamics or relaxation with the probe pulse, which is sent at variable delays after the pump pulse. In fact, we can detect several things. We can detect the light going through, we can detect charged particles created after the interaction with the probe pulse, and so on and so forth. And in fact, we don't care if our detectors are extremely slow, because what is dictating our time resolution is the duration of the light pulses we are using. So as we said, again, the duration of the light pulses needs to be as short as the dynamics we want to explore. So in fact, the progress in the development of laser technology or laser sources has opened the possibility to access faster and faster mechanisms. As you can see in this very nice graphical representation of the briefest events ever measured along the years, we can observe a very nice correlation with the progress in the laser technology. As you can see, there are some points in which this has been drastically improved. For instance, at this point here, there's been the discovering or the introduction of the chirp pulse amplification technique, which I don't have time to go through. This has been a Nobel Prize in physics, and it has allowed indeed to shrink very consistently the time resolution of our experiments, since the laser sources could have been brought towards shorter and shorter pulse duration and access ultimately the femtosecond domain, and therefore it has been possible to observe in real time, for instance, the breakage of a chemical bond, or in fact the chemistry in action. This was at some point reaching a saturation again, and until the introduction of the Attosecond technology, which indeed has allowed to explore now the electronic motion in matter inside atoms at the beginning, and then molecules or more and more complex targets. So since 2000, the introduction of this Attosecond technology has quite revolutionized the time result spectroscopy and given access to novel phenomena. Attosecond light pulses are not available out of conventional laser sources. In fact, laser sources can deliver femtosecond pulse durations, and this is in fact also intrinsically limited by the carrier wavelengths of these sources and various other aspects. So the only way to obtain shorter pulses is to shift the carrier wavelength from the near infrared, which is the typical carrier wavelength of for instance, the titanium sapphire laser, to the extreme ultraviolet or the vacuum ultraviolet. And this is done exploring an extremely nonlinear process, which is called ion-harmonic generation. I will dive into it in a moment. But basically, this nonlinear optical process is based on the idea of achieving very high laser intensities, in fact, above 10 to the 14th over square centimeters, that you can reach by focusing a laser pulse into a gas target, for instance, an atomic target. And then to drive these nonlinear optics beyond the perturbative limit to obtain radiation at higher photon energies and with much shorter pulse durations. The process has been already mentioned in the previous presentation by Professor Capasso. I would give you a little bit more details about it. So it can be nicely described with a semi-classical model, which is actually very accessible also by non-experts. It has been introduced in 1993 by Paul Corkum. In fact, when you focus an intense laser field over, for instance, an atomic target, the amplitude of the electric field of the laser is now comparable with the electric field experienced by the electron into the Coulombic potential. The Coulombic potential is therefore deflected, it's deformed by the presence of this dressing laser field, and the electron can now tunnel out from the parent atom. This tunneling ionization is in fact the only quantum step, because everything else can be nicely described classically. In fact, now you have a free electron in the continuum, which experiences the presence of a laser field, and therefore it is accelerated. The laser field is oscillating, so the electron, which is ionized, is accelerated in the continuum far away from the atom, and then it's driven back by the laser field. And then there is a non-negligible probability of recombining with the parent ion, and via this recollision event, the excess of energy which is gained by the electron in the continuum is released as a form of a photon, which is now a high energy photon, as I mentioned before, in the extreme ultraviolet region, and it has a very short duration. The typical emission is something like this. So you have discrete peaks, and it exhibits a cutoff frequency, so it doesn't extend forever. Of course, there's a maximum photon energy that we can reach, and this depends over several parameters, including the laser intensity and the laser driving field wavelength. Why we get shorter pulses? Well, every half cycle of our infrared driving laser pulse, we have this three-step mechanism, so we can emit a very short XUV pulse. This is happening in half a cycle, as I was mentioning, but the laser field is composed by many half cycles, and therefore we end up with the train of attosecond pulses in the time domain. These attosecond pulses are separated by half of the period of the laser field, as I was mentioning, and this in the frequency domain, in fact, nicely corresponds to a comb of odd order harmonics, which are then separated by twice the frequency of the driving laser. So this is a pulse train, so it's nice, but it's not yet a single attosecond pulse. In order to achieve a single attosecond pulse, we can apply a trick. We can, in fact, play with the polarization, for instance, of our driving field, as such that we can confine the ion-harmonic generation process only to one half cycle of the driving field, and this would now correspond to a single attosecond pulse that in the frequency domain has a very broad emission, which indeed has no energy selectivity, but a very nice temporal structure. So nowadays, there are several attosecond sources that have been developed, covering mostly the VUV region, but also the soft X ray region is now accessible with attosecond sources with photon energies covering the whole water window, extending up to 500 eV and beyond. And the shortest attosecond pulses are in the order of a few tenths of attoseconds. You can also find here a very nice review paper covering all the most recent technological developments on attosecond sources, which I will not have the time to cover in this lecture. So now we have this XUV attosecond pulses, but we want to use them for time result measurements. As I was mentioning, this is an extremely nonlinear optical process, and therefore it has also a reduced conversion efficiency. So it's not easy to get high photon flux, at least not comparable as the photon flux you can obtain from free electron lasers. And therefore, we often don't split the XUV attosecond pulses in two arms to perform an XUV pump, XUV probe experiment, but we combine our XUV attosecond pulses, for instance, with a residual part of the IR laser, so that we have an XUV pump IR probe setup. Here you can see a typical interferometer. This is completely developed in vacuum, as the XUV light needs to be transported in vacuum. And you can see here also the typical characteristics of those pulses. So in our case, we generate attosecond pulses covering a spectral range up to 40 eV, and we combine them with sub-4 femtosecond infrared or near-infrared pulses for our experiments. And the duration of our attosecond pulses is in the order of a few hundred attoseconds. Now, using this technology, our goal was to try to investigate it, as I said, get access to ultra-fast electronic mechanisms. And in particular, my group has been involved in various activities in this direction, but today I would like to focus with one specific investigation, which is the investigation of charge migration occurring in molecules. Charge migration has been at first only theoretically predicted by the group of Lenz Sederbaum at first, and then other groups, including Françoise Remacle, indicating that after a sudden ionization or excitation of a relatively large molecule, in fact, you can create a non-stationary charge density distribution. And non-stationary means that the electronic charge will start to move very rapidly in the molecular backbone, even before the nuclei have the time to rearrange. Here is a sketch of this mechanism where you can see a calculation performed in a small peptide. An electron is suddenly removed, and then you see that the charge density distribution is not static, but as I said, rapidly evolving. You can see the charges going from one side to another of the molecular backbone within a few femtoseconds. And this can also happen within a few hundreds of attoseconds, depending on the energy separation between the electronic states. In fact, what you need to do is to create a superposition of electronic states, and this can be done either by ionizing an electronic correlated state, which is by definition a superposition of states, or by using a broadband laser pulse, which is intrinsically creating a coherent superposition of electronic states. This process, as I said, occurs very rapidly, and so we need attosecond or few femtosecond light sources to get access to it. And it has attracted our attention, as we know electrons are playing a major role for the creation of bonds or the cleavage of bonds, and therefore the main question was, can we use these ultra-fast mechanisms to ultimately control the reactivity of the molecule? Or in other words, can we achieve a charge-directed reactivity? So the way we approached this problem was actually to implement a relatively simple experiment at first. And we studied the photofragmentation in a time-resolved manner of aromatic amino acids. In this case, this is the amino acid phenylalanine, which are the nano-sized antenna of our proteins, as they can capture very efficiently light. And here we were sending a sequence of an XUV attosecond pulse, followed by a few femtosecond infrared pulse, as I was describing before. And in fact, after ionization, the molecule fragments in many pieces, and you can measure with the detector the mass spectrum as a function of the delay between the two pulses. This is the typical fragmentation of the molecule. So you have a high level of fragmentation with several fragments as the deposition of energy is relatively high. And in this very rich mass spectrum, we have been interested in a very small fragment, in fact, the doubly charged ammonium ion. And by tracking the mass of this fragment as a function of the delay between the two pulses, we could clearly identify rapid oscillations, as you can see here, in fact, happening within a few femtoseconds, both for the phenylalanine amino acid at first, and then later on in another experiment in the tryptophan amino acid. Now, those oscillations were too fast to be assigned to any of the vibrational motion of the molecules, and therefore we collaborate with our theory partners at that time, Fernando Martin from Universidad Autónoma de Madrid, to find an interpretation or to find an explanation for those fast oscillations, whether this was a signature of the charge migration. And indeed, we could finally understand that that was a signature of charge migration. And in fact, those oscillations could be assigned to the migration of the charge from the amino group to the carboxylic group of the molecule and back, occurring on a 0.25 petahertz scale, so few femtoseconds. Same was for the amino acid tryptophan that was analyzed later on, and here the frequency that we observed in the experiment was the result of the interference between two oscillatory dynamics, one from the amino group to the ammonium group of the molecule and back, and the other one again between the ammonium and the carboxylic group of the molecule. That was very fascinating. And we then therefore decided to investigate other molecules which are biorelevant, or again, building blocks of our big biomolecules. In this case, DNA building blocks. We studied adenine as we know, again, damage of the DNA can occur both for exposure to UV light, but also to high energy photons or even secondary electrons created by high energy radiation propagating in the human tissue. And in this case, the deposition of energy, for instance, during phototherapy treatments, is in the order of 20, 30 eV, so exactly the energy that we have available with our photon sources. So we were looking already at the photostability of the molecule when exposed to such an amount of energy. And again, you can see that the mass spectrum reported here show a high level of fragmentation, about 80% are fragments, as expected. And again, we were looking at the mass spectrum of adenine when launching IR probe pulse after the XUV photoionization. And there we could clearly see that, in fact, when you look at the pump probe signal, this is the integrated mass of each individual fragment. In fact, the larger fragments are drastically reduced at 0 time delay. So when we send the infrared pulse synchronized with XUV pulse, and the smaller fragments are increasing. And this is basically due to the fact that we deposit a further amount of energy using our infrared laser on top of the XUV pulse. And this is bringing the molecule to further excited states that ultimately leads to a higher level of fragmentation. However, a very interesting observation back then was the presence of the detection of a stable molecule. So this is the intact adenine molecule, doubly charged, which is not showing up when we send the near pulse synchronized with the XUV pulse, but only 2.3 femtoseconds afterwards. So we need to wait for a very short time before we can observe the creation of such a stable and intact adenine molecule. And this time, again, it's too short to basically hidden or to basically have behind a structural reorganization of the molecule. So we were looking again for an electronic mechanism to explain this sort of stabilization mechanism of the molecule, which otherwise undergoes photofragmentation. And here we came up with this simple explanation in collaboration with our theory collaborators again, the group of Angel Rubio this time from the Max Planck Institute of the Structural Dynamics here in Hamburg. In fact, after the ionization from our XUV pulse, we have an electronic process happening, and this is due to the correlation of the states and the fact that an electron is rapidly filling this vacancy by promoting a second one to the outer valence. This shake-up mechanism, in fact, allows us to have an electron available now in the outer valence for our probe, near infrared probe pulse, to be ionized and therefore create this stable and intact adenine. Now, the interesting aspect here, in fact, is that theory predicts that during this shake-up mechanism, which is again due to the fact that we create a superposition of states by ionizing a correlated state, we don't have a static charge density distribution, but we have a migration. And the migration in this case is an out-of-plane inflation of the charge. So here you can see that the charge is in fact rapidly inflating, and this inflation is responsible for an increased absorption of our IR probe pulse, which otherwise would not occur. But we need to wait around 2 femtoseconds before this inflation is effective and in fact allows us to absorb the infrared probe pulse. So this is just an example how we can use the charge migration mechanism to obtain a new outcome for the molecule, in this case, the stabilization of a molecule which otherwise would be photofragmenting after the exposure to UV light. I showed you examples of the study of charge migration in photoionized molecules, and this inevitably happens every time we use VUV or XUV light, as this is above the ionization potential of all the molecules. And this radiation is, in fact, very interesting, but not representing, of course, the majority of the phenomena which are happening on Earth. As we know, ionizing radiation is not penetrating our atmosphere. And a very interesting wavelength is, of course, the UV photon energy or UV light, as it's responsible of many photoactivated mechanisms. We have seen before, I gave you the example of the photo-induced damage or recovery, but also UV or visible light is very interesting for optimizing light harvesting and getting, of course, energy out of the solar light. So what was the effort of my group in the last few years was actually to push the attosecond technology forward and, in fact, combine our attosecond pulses with UV light. And, in fact, we wanted to preserve the temporal resolution of our, so to say, experimental setups by generating extremely short UV pulses. And in order to do that, we have been exploiting the third harmonic generation in gas. We have designed special gas cells in glass via a laser micromachining of glass in order to confine the gas efficiently over a few millimeters. And these special gas cells, which allow basically to achieve very high pressures with an optimal gas confinement, allowed us, in fact, to reach the sub 2 femtosecond regime for the generation of the UV pulses. And this has been published in 2019. In fact, back then was the world record for the shortest UV pulses ever generated. Now I want to provide you an example where we can use these ultra broadband UV pulses, in fact, to initiate charge migration in a special class of molecule, in fact, to control their functionality at the electron time scale. This class of molecules are chiral molecules, and I would like first to briefly introduce the concept of chirality. As many of you know, chiral objects are non-superimposable mirror images, one of the other, and the easiest example, of course, is our ends. And in the case of chiral molecules, the two enantiomers, this is how they are defined. They have the very same chemical composition, but being one mirror image of the other, they have very different chemical functionalities. And for instance, all the living organisms are mostly dominated by one of the two enantiomers, for instance, the L amino acids, and therefore homochirality has emerged, so to say, in life. And in the field of pharmacology, chirality is very important because a molecule that has an healing effect for our body has a corresponding enantiomer that has a very toxic effect. For instance, the case of methamphetamine is the most popular, where L-methamphetamine is a very popular nasal decongestant, while the D-methamphetamine is a very dangerous drug. So chiral recognition is in fact a very important topic, and in our case, we didn't want to really work on implementing or introducing new chiral recognition techniques, but in fact to look at the chiral response of molecules when electron dynamics is activated. But in order to do that, we need to exploit a method to look at the chiral response of the molecule. There are different methods. One method is the most, so to say, popular method is to let the chiral molecule interact with another chiral object, for instance, circularly polarized light. And for instance, one can have a look to the circular dichroism, which is the differential absorption of light when it's left or right circularly polarized. And this is actually quite a weak signal, as it is based on interactions between the electric and the magnetic dipole transitions. There's another effective method which has been introduced, which is the photoelectron circular dichroism. And here you use circularly polarized light to ionize a chiral molecule. And here, by keeping the same molecule but changing the polarization of light from right to left, you can see that the photoemission is happening forward in one case and backward in the other case. So looking at the asymmetry in the photoemission, one can detect the chiral response of a molecule. In our case, we were studying methyl lactate, which is a derivative of the lactic acid. So it's a molecule which is very important for our muscle metabolism and also has quite a high ionization potential. Therefore, it's very difficult to ionize it, as we wanted to prevent ionization of the molecule, but simply excite it or excite this coherent superposition of states using our broadband UV pulse. So here you can see the very broadband nature of our UV radiation, and we use a two-photon transition to excite a coherent superposition of Rydberg states. Afterwards, we use a circularly polarized infrared field to probe the chiral response, and we need only one photon transition to go above the ionization threshold and produce a photoelectron. So here is the sketch of the experimental setup. We can see a sequence of UV, linearly polarized exciting pulses and circularly polarized probe pulses, both with a very short duration. And then we looked at the electrons this time, not the ions, emitted after the ionization from the IR probe pulse. If you look at the angular momentum distribution of these photoelectrons, you can find quite a boring map. The interesting things come when you calculate the PECD, which is the difference between the circularly left polarized measurement with the circularly right polarized measurement. And here you can clearly see the emergence of the asymmetry I was mentioning before. So if this is the laser propagation axis, you can see that for one enantiomer, the emission is mostly backward, and for the other enantiomer, the emission is mostly forward. So we can clearly detect the presence of chiral molecules. But now these are static measurements, so nothing fancy. The fancy thing starts when we look at the time resolved measurements. So we look at the effect of the electron dynamics. And now we can see that, in fact, the PECD is changing very rapidly in a scale of a few femtoseconds. You can see that, in fact, we change the emission in certain energy regions from backward to forward, backward and forward again. So we can really switch the chiral response by looking at the evolution in time. You can do a more quantitative analysis, analyze via the Legendre polynomial and extract the beta parameters. This is a bit technical, but in fact, what you extract is again an oscillatory dynamics. In the PCD, this is extracted for various kinetic energy ranges of the photoelectrons, and we can clearly see a beating frequency and the switch off the sign that has been observed in the PECD. Finally, theory helped us again in understanding the role of the electronic coherences. We could, in fact, via a collaboration with Bernard Pons from CELIA, see that with our UV pulse, indeed, we create different coherences. The 3D 4P coherence is responsible for the beating in the low kinetic energy region of our electron spectrum, while the 4P 4D coherence, which is created in the molecule, is responsible for the oscillatory dynamics in the high kinetic energy regions. These coherences are in fact activating the charge migration, so the electron dynamics, which is ultimately modulating the chiral response. So the message here is that we could see a completely switching sign over the chiral response of the molecule to chiral light without switching enantiomer on the electron time scale. So to just conclude and give you some take-home message, so to say, we can generate nowadays light pulses which are as short as few tenths of attoseconds. Attosecond pulses are now routinely produced with tabletop sources, as I was showing to you. But of course, X-FELs are also progressing very rapidly, and now attosecond pulses are available also at free electron lasers, and I'm pushing Attosecond Science towards the X-ray domain. We can track pure electron dynamics or charge migration in relatively complex molecules, and I showed you that we could take advantage of the electron dynamics to control certain molecular functionalities, for instance, the chiral response, which ultimately could lead to a charge-directed reactivity. With that, I would like to thank you. Of course, all my group, these are all the students who contributed to this work. You can visit our website to see our other activities, and thank you very much for your attention. I'm now ready for questions.